Thank you. Good morning. I hope everybody is feeling good after that wonderful dinner, drinks, and entertainment last night. That was quite a show. So welcome to this morning's session. And Gary Atlin from the Bill and Melinda Gates Foundation will kick off this morning's session and talk about what do we need to do to bring genomics tools to routine cultivar development. Take it away, Gary.
Sorry? It's probably this one? Can you hear me? Okay, so we'll do that, I think. We'll just get this, yeah. Okay. Well, thanks very much. Thanks to Rajiv for inviting me to participate in this very useful meeting, which is really becoming a crossroads for breeders and biometricians, geneticists, and people who are interested in increasing the effectiveness of breeding for our targets. I'm going to quickly run through three basic areas. Applications of high-density marker information in cultivar development as opposed to genetic analysis. A number of these applications have already been discussed in this meeting, and so I'll touch on them. I'd like to, because I have the microphone, give you some of my opinions on genomic selection, and we have a very nice series of talks following me on aspects of genomic selection by the best people in the field in plants. So if I make any mistakes, there's plenty of opportunity for them to be corrected. And I'd like to finish off with in terms of talking about what really needs to be put in place to enable people doing cultivar development to benefit from and exploit high-density genotypic information, talk about the enabling environment that we need to develop and the responsibilities in particular of research managers and donors. I speak with two hats, one as a plant breeder, which I was for 30-odd years, and one as a person working for an organization that funds a lot of plant breeding. And it will be completely unclear which hat I'm wearing at which moment. I'm sorry. Genomics applications for cultivar development and the needs of plant breeders are somewhat difficult and different from those required for genetic analysis. And most of the discussion on genomics applications tends to focus on genetic analysis. But a lot of the issues that breeders face are somewhat different from those faced by people who are essentially doing gene discovery work. Selection decisions breeders make selection decisions, first of all. So they're concerned with issues of selection bias. So are geneticists, but they often don't realize it. And they need to make their decisions very quickly in a narrow window of time. So they need very robust, user-friendly pipelines, and the haplotype effects that they measure or seek to deploy are very dependent on both the genetic and environmental context. We've heard a fair bit about that over the last couple of days. So they need to be able to, they need very good information on what effects are going to look like in different backgrounds and different environments. They also manage large and constantly changing and growing data sets. And in fact, the management of these databases and their development is, in my opinion, the key problem that we face in applying genomics. Not the pipelines, not the methodology at this point, but actually getting the databases into a shape that they can actually be exploited. And breeding strategies are highly dependent on costs. So genotyping costs, line development costs, and we have a couple of experts on that issue here. I encourage you to talk to them about them. Jeff Endelman, John Hickey, Aaron Lorenz are doing the best work in plants on trying to work out actually how to use high-density genotypic information in a cost-effective way. The breeding applications for high-density genomic information, the primary ones that I'm aware of include quality control genotyping, tracking high-value alleles in forward selection, and line conversion. Then there's two genomic selection applications which to my mind are often confused. Predicting the phenotypic value of selection candidates, hopefully more cheaply and accurately than you can via phenotyping alone. And selecting parents for the next cycle of breeding and that's called genomic selection, but it's not what a lot of genomic selection, for instance, in animal breeding, where the idea was developed, attempts to do. Selecting parents for the next cycle by genotype alone to reduce cycle time. These all require a genotype, and I think to minimize costs, they should be done out of a single DNA sample. I was told by a young person that IMHO means in my humble opinion when I got an email. So I'll use that sometimes to indicate that this is my opinion, and I realize that it's not, it's context dependent, but I think this is the way we're going. In terms of quality control, modern breeding programs really must include a quality control genotyping step. Anyone who starts genotyping all their parents realizes that misidentification rates are much higher than expected. Quality control genotyping pays for itself by eliminating the costs, many times over usually, by eliminating the costs of advancing and testing incorrect populations. I have done this myself, advanced very far a bad population. And, it's a lousy feeling to 2 years down the road realize what you've done. I myself, I've made every conceivable mistake. There's really a question of what stage in the breeding process you should use the quality control genotyping. I think it's anything now, with the costs that we have for relatively high densities coming down with GBS to $10, $15 a sample very, very quickly. Anything that goes into replicated field testing should be genotyped. This requires roughly 100 high information content SNPs, which as uniplex assays from LGC would cost about $12 per sample. And this is really converging on what you could get for the same amount of money, but with 100 times at least the marker density with GBS. Which can be used for many purposes other than quality control. As part of the Gates Foundation's efforts to support breeding programs in an improvement process, we will really be asking partners to implement a quality control system. So, and by implementing a quality control system, it means having procedures for quality control, having the data in a database so that it's easily accessible. Another application is tracking high-value haplotypes, QTLs with large effects. Breeders want to know if individuals carry specific alleles of interest. At the moment, wheat breeders, I don't know how many alleles they would want to track, Susanna or Sukhwinder. Something like 30 or 40 loci now would be for quality, for disease resistance. Would that be in the right ballpark? So they need to know the identity by descent status of all those loci. And assays detecting specific target sequences are generally, preferred by breeders because of their simplicity of interpretation. But they're expensive and single purpose, and with the same DNA sample, you could get you could type the whole genome. For this to work, however, to use GBS to track identity by descent for the key alleles that you are interested in, you need to have very reliable pedigree information associated with that entity and the genotype. I think that, again, we want to use a single assay for this purpose and for quality control in most situations, in my opinion, unless the allele is an absolute prerequisite, and therefore you can screen very early in the program, and only a few loci are required. The ability to track the provenance of an allele is an absolutely critical capacity of a breeding program. So again, Gates will be encouraging and trying to support our partners in implementing breeding databases like the tools provided by the integrated breeding platform and other systems. Moving on to other applications, genomic selection type applications. We've heard discussion of applications that really amount to predicting the phenotypic value, for lack of a better term, how an individual selection candidate is going to perform in the field more accurately and cheaply than via the phenotype alone. And I would, not to coin a phrase, but call that genomic selection for phenotypic value for the sake of this discussion. This, at the moment, is the most commonly tested and promoted application of genomic selection. Selection response is a function of selection intensity, selection accuracy, and the genetic variance. And for phenotypic selection, selection accuracy is just the square root of heritability. This accuracy is potentially limited, I think, by non-additive gene action, and there are prediction methods that apparently accommodate those. But this genomic selection application is essentially a method of increasing selection intensity. And to increase response to selection in this way, the gains from increased selection intensity have to outweigh losses from reduced accuracy relative to phenotypic selection. There are some problems with that. Gain, as you know, gain from increased selection intensity is far from linear. Roughly speaking, to double selection response, you need to increase population sizes tenfold. Now, multinational seed companies like Pioneer, Syngenta, Monsanto are doing this in their breeding pipelines, but they can do it because their line generation costs and genotyping costs are extremely low. Another problem with using this approach is that phenotyping plants for yield to a repeatability of about 0.5 or even 0.7, a heritability, a square root, an accuracy of 0.5. Costs, in my experience, about $200. If you figure $25 a replicate, 4 locations, 2 reps. That would be something like that. A little, with a little there for line generation. Whereas in most species, line generation and high-density genotyping costs at the moment are at least $30. So if you multiply that by 10, that's the sort of, if you have a very accurate genomic selection protocol, and you want to double rates of gain in this way, you need 10 selection candidates for every one that you move into the breeding pipeline. That's expensive, and the accuracies have to be very high, and the line generation and high-density genotyping costs very low, and this has been worked out by in a number of very useful recent publications. Genomic selection accuracies for yield are not really high enough for this application to work, in my opinion, especially for non-relatives, and I think we're going to hear more about that this morning. For relatives, genomic selection seems to add relatively little accuracy to pedigree-based BLUPs when there's substantial structure in the population, and a lot of the initial experiments in this field have been the results have been biased upwards by structure effects. For example, if you look at an analysis of in a maize mapping population, association mapping population, in a cross-validation study that gave an accuracy of about, the other issue is that prediction accuracy across groups tends to be quite low among non-relatives. And if this isn't taken into account in the cross-validation experiment, you can get a very high prediction accuracy that doesn't hold up when you try to do validation across groups. Training and the take-home message is that training and validation populations have to be very closely related with similar allele frequencies and linkage relationships, and we'll, I believe, hear more about that from Aaron. And databases for, and the upshot of this, I think, is the databases for breeding programs implementing genomic selection have to be able to automatically subset out only close relatives for model training. And I think that's, that essentially has to be inbuilt. And that type of query. Pancho Crosa has just published a very interesting comparison of the accuracy of pedigree, phenotypic selection, and BLUP with in a set of about 500 maize doubled haploid lines. Apologies, Pancho, if you're going to show this again this morning, but you'll do it in more detail, I'm sure. Where this gives you an idea of the additional value of adding the marker information to good pedigree information in predicting a number of traits. This is one of the best examples of this type of an experiment on genomic prediction accuracy that has a proper control, that is control against the accuracy of using the pedigree alone. There is a benefit, but it's relatively marginal, and getting the pedigree information into breeding applications is clearly important. For applications of genomic selection within populations that rely only on Mendelian sampling, we are looking at accuracies. There have been a number of CIMMYT biparental populations in which this has been estimated, and the accuracies, similar to other studies, tend to top out at, even with relatively small, low marker density, as was the case in this experiment, conducted by the CIMMYT maize program and analyzed by Pancho Crosa's team and Vanessa Windhausen. They tend to top out at about 70% of the correlation based on phenotyping alone. Breeding applications that involve selecting parents for the next cycle, these accuracies are probably quite adequate for that, even within relatively small training sets. So this type of Holstein-type genomic selection breeding plan, where you form a narrow population and rapidly cycle it, looks reasonably promising to me. I feel that that's the area, one of the areas we should be focusing. The accuracy is only adequate, however, I think is likely to be adequate in very narrow populations. And the training sets will have to be population specific. And repeatability in the training sets will have to be very high. This means that the phenotyping load for each training set will be high. And we had experience with this in the CIMMYT maize program, that if you begin a number of MARS or population-specific genomic selection programs, the phenotyping load can very quickly become extreme and displace effort from the breeding pipeline. Just to wrap up this discussion on applications of high-density genomic information, there are large payoffs immediately available from quality-controlled genotyping, and any well-managed modern breeding program should be doing this. The costs seem to favor a single assay for all genotyping needs if pedigree information is well managed, and so every breeding program needs to manage its pedigree information well. Genomic selection to predict the line phenotype is a selection intensity-based strategy, and current genotype to phenotype cost ratios for small programs make this application unpromising. It might be more promising if we focused on adaptation-related traits. Or quality traits that should be easier to predict at higher accuracy, and would prevent us from moving to the starting line for field testing materials that are not going to be useful. For genomic selection to select parents for rapid cycle improvement of closed populations, I would say we need to continue working on this, but to proceed with caution because of the expense and phenotyping load that this can impose on a program. Just another word about those logistics. Experience at CIMMYT to date with over 40 rapid cycle genomic selection populations indicates that this is quite difficult to integrate into a breeding program. At first, public programs almost never have the coordination level to execute. There will be mistakes. It's a specialized function that should not be loaded on top of the line development program. It has to be done separately. Has a very high phenotyping load and should be started with a relatively few closed multi-parent elite populations. And the prerequisites for using this type of information and this type of application are really high quality phenotyping, as everyone knows, integrated databases for pedigree phenotypic and marker data. The query capacity of the database needs to permit rapid isolation of groups of relatives with a defined coefficient of parentage, and very good prediction and support, decision support pipelines, but the database is really more important at this point than the pipeline. I often trot this out. It's basically a Holstein breeding plan for plants. I think it is worth looking at, particularly in the context of a large breeding institute like or a public program, where you set up an elite, closed, high LD population for rapid cycle marker-only selection, extract lines. In this case, we're talking about maize, so double haploid lines, genotype them, and provide them along with a GEBV to, say, different companies. Each company gets its own proprietary set of lines, phenotypes, and returns the phenotypic data for model training, but retains whatever lines it wants to commercialize. The advantages of this, so small companies can benefit from this rapid cycle approach, but the complex work of managing the populations and prediction is done by a central organization. So advantages include that small programs can access haplotype effects. They can essentially share haplotype effects without sharing germplasm. Partners benefit from the phenotyping done by other network members. Lines are proprietary. Only the the models are shared. The hub programs allow economies of scale to be enjoyed by small seed companies and partners. I think that this type of model can really provide small companies and national programs with powerful breeding technologies now only available to multinational companies. And serving as the hub for this type of network, I think, is potentially an important new role for CG breeding programs. But cultivar development programs can only exploit high-density genotypic information to increase rates of gain. I think if we start to be much more serious about research management in our systems, breeding is really product development, not research. Research managers need to accept responsibility for delivering high rates of genetic gains. This means taking responsibility for defining breeding targets, setting performance goals for programs, and setting genetic gains targets relative to widely grown checks. Taking responsibility for ensuring that basic tools of modern breeding are both available to researchers and that they are used. Managing teams for product delivery and to some degree de-emphasizing publication as the primary measurement of a scientist's productivity. The investments needed for systems change leading to higher rates of genetic gain in farmers' fields include, on the technology side, engineering support for mechanization, automation, digitization of field operations, support for breeding programs to upgrade research stations into world-class phenotyping hubs, support for adoption of breeding information management systems and databases that allow rapid analysis and decision-making for selection, consultancies to help breeding institutions optimize their pipelines and develop their databases. And the CG really needs to lead in these areas. In many cases, it's not. It also requires investments for systems change leading to in the area of management, capacity metrics, and incentives. So in terms of upgrading skills of professionals to use methods that they weren't trained to use, enabling and informed management with clear goals and that take responsibility for gains in their programs. Use of routine intermediate metrics that are already used in the private sector in terms of repeatability and data quality, program size, advancement systems. An accountability system that models good management and rewards national and international programs, and we're doing a lot of work on that at Gates at the moment. And from the donor side, sustained support for systemic transformation of breeding programs and longer funding cycles will be needed to implement this modernization. Without going into detail, because there's no time, I'll just note that the Gates Foundation is developing a breeding program assessment tool or scorecard that will help public sector breeding programs launch their continuous improvement efforts to achieve higher rates of genetic gain. This will involve a questionnaire covering most important areas of breeding program operation and management, about 70 questions. And the way the questionnaire is structured, it really lays out major management issues in cultivar development. It covers areas including breeding program organization, infrastructure, field breeding methodology, plans for cultivar development and commercialization, support for the product once it's released, measurement of impact of the program, strategic planning and management, including breeding goals, budget management for performance, and staff and team performance evaluation. We're incorporating these prerequisites and metrics that are modeled in the assessment into our own grants via both the agreements and the results frameworks of the plans. And these prerequisites for support would include commitments to modern database and decision support, seed storage, off-season generation advance, capable staff, support for program leadership, the support of program leadership, having clear product concepts, having a clear target environment, having a plan for improving the pipeline, and a plan for disseminating the product. These don't all have to be in place to get our support. The plan to do these things needs to be in place. Metrics will include measures that are often used in the private sector, including measures of program size, advancement rate, testing effort, repeatability, data return rate, and direct measurements of rate of gain. We would hope to support increased rates of gain by essentially, in sub-Saharan Africa and South Asia, by sort of a three-pillared approach where we try to help breeding programs understand management for increased effectiveness using by providing them with scorecards and tools that they can monitor their own progress with, co-funding agreements with research organizations for improvement plans to reach the standards, and consultancies to support the planning and implementation of improvement plans. To sum up, for the type of systems change and technical innovation that we need to increase rates of genetic gain, I would say that the first is improving the standards of management. Second would be pipeline and network optimization through mentoring and consultancies. The third would be application, getting our breeding information under control and really into the hands of breeders. And improvements in phenotyping, basic marker applications like quality control and tracking IBD alleles, and then high-density genomics. A high rate and sustained rate of genetic gain is a key component of agricultural transformation. The rate of gain delivered to farmers is the key measure of effectiveness of a crop improvement system. The public crop improvement system is lagging somewhat in technology and management methods, and I would say is underperforming. Research leaders, scientists, and the donor community need to take responsibility for improving this state of affairs. The CG programs, national programs, breeders and small companies in Africa and South Asia need sustained support to apply basic modern breeding technology and optimize their pipeline. And the CG system needs to grasp a leadership role in supporting modernization of the international public breeding system. And the Gates Foundation is committed to help with this process. Thanks very much. And sorry for going over.
Yeah, you weren't too much over. Anyway, in my humble opinion, that was a very lucid and comprehensive review of sort of the current state of what how the foundation and I think many breeders would view the way breeding programs need to be organized. So thank you, Gary, for that. Is there any questions? Yes, Roberto.
Have you thought about the problem of EDV, essential derivation? I mean, this may not be much of a relevant problem in certain types of agriculture, but have you given a thought to that?
Well, they come out of the same, it's just, it's not essential derivation in my opinion. I don't know what the EU rules would be.
I think it depends what sort of standard crop.
Selection out of essentially a synthetic or a randomated population is quite different than back-crossing and making a tiny substitution.
No, I understand that, but it depends where you set the threshold. Because
sure.
if you leave it to some seed companies, they want to bring it pretty low.
Yeah, I think that's manageable, and I don't think it's much of an issue in this context.
Yeah, one more question, Graham.
Gary, I don't know if this question is for you or Pancho actually, but to get away from all the big ticket items and ask the interesting question for a bit, what is the prediction ability if you don't use pedigrees and only use markers? Because the excuse I've often had for sidestepping the database issue is that markers will do it all.
Well, I mean, the simple answer to that is they won't. You know, if...
Anybody quantify?
Yeah, I mean, what will happen is what you see in the cross-validation experiments when you remove structure as a source of accuracy. When you do that completely, you see huge drops in accuracy and you know, the relatively marginal increases. So, you know, if you essentially operate your breeding program as if it were a cross-validation experiment or a GWAS experiment and try to predict from last year's, those, you know, those, you'll predict what groups, you know, what families the material came from, and that's all you'll get. You'll get no benefit or very little benefit from Mendelian sampling. Yeah, I don't think so. I mean, Jesse may or others may have other experience, but from what I've seen so far in the CIMID program, that's the case.
So maybe if we have time, we can follow up some more on that. Thank you, Gary, very much. Nice presentation.